everyone. Today's presentation will explore a recent study in the field of molecular biology by Van Bergman and colleagues, which looks at elucidating shark diets with DNA metabarcoding from cloacal swabs. We will explore the study conducted by Van Bergman and colleagues by first presenting background material, expressing the hypothesis and predictions, going through the research design and methods, assessing the results, and concluding with a discussion. We'll also discuss the relevance of this research to the area of molecular ecology, as well as potential future applications. The introduction of this study brings light to the fact that when analyzing the diets of large aquatic species, such as sharks, the previous methods are labor-intensive or harmful to the animal, since feeding behavior is hard to directly observe. The most common way to assess diet is through lethal sampling and invasive stomach aversion. This method works relatively well, but is bad for threatened species and is challenging because it is labor-intensive and the food in the stomach is often already digested and unrecognizable. Another method is to use biochemical tracers, which require extensive sampling of both the predator and prey. This can be difficult as most sharks feed across multiple trophic levels, so species-level resolution of the prey often remains uncertain. A new upcoming application of high-throughput sequencing in conjunction with DNA metabarcoding is working well in diet reconstruction. It allows for high-resolution species identification within the stomach contents and fecal samples. However, it can be difficult to get fecal samples because defecation is rarely observed, and once the feces enters the water, environmental DNA, or eDNA contamination can happen. If contamination does happen, it becomes hard to tell if prey DNA is from the feces or the eDNA. Therefore, the solution is to find an accurate, efficient, minimally invasive method for collecting and metabarcoding fecal DNA, or fDNA, while reducing eDNA contamination. Because sharks remove both solid and urinary waste through the cloaca, there is an opportunity to sample fDNA directly from the cloaca. This can be done soon after defecation using cotton swabs, and then metabarcoding the fDNA from the samples. There were two main objectives of this study. The first objective was to determine whether known diet can be identified by fDNA metabarcoding and if eDNA contamination can be excluded. This was done by putting focus on a single primer set that targets the 12S ribosomal RNA region to identify a prey not in the natural diet of the sharks. The second objective was to test the usage of this method on free-range bull sharks, using the same primer to test if the method can be used to identify prey signatures consistent with previous diet studies and available prey. When completing the controlled part of this experiment, there were a few predictions. The first prediction was that the DNA recovered from the cloaca sample would be only from the predator and known prey, and not from any other free-floating eDNA. Next, they predicted that the prey DNA would be detected only after feeding to ensure there is not eDNA contamination. And finally, that eDNA contamination was not a source of prey DNA detected on the swabs. We'll now examine the study's research design and methodologies. For the laboratory conditions, first 3 male juvenile sharks were caught using netting, rod, or reel fishing in a tidal stream system on the coast of Cape Eleuthera, Bahamas. After being caught, the sharks were exposed to ambient seawater conditions with a natural photo period for 14 to 15 days. To reduce meal retention time and speed up feces production, individual sharks were fed 6% of their body weight in fillet. Then cloacal swab samples from the place of capture were obtained every day for fDNA analysis. Sharks were held upside down in a tonic immobility with the pelvic fins torn apart and the shark's posterior half remaining above water in order to obtain this information. Then, to test if the cloacal swabs were contaminated, the eDNA species composition in the surrounding seawater from each of the water samples where the sharks were housed were examined as well. Sharks were similarly caught and a cloacal swab was taken, as well as their sex and length measurements when the study was applied to a field population in the Shark River estuary in Florida. The target genetic marker under investigation was a 171 BP fragment of the mitochondrial 12S rRNA region, which was used to amplify fDNA and eDNA from the cloacal swabs and water. Now we'll take a look at the study's findings. Figure 1 is laid out in a very interesting manner, depicting the study's timeline with a focus on the controlled feeding component of the research. The first day of the study refers to when the shark sample was taken and when the cloacal swab collection began. Day 3 then marked the beginning of the shark's controlled feeding, which lasted every day until the trial's conclusion, which was approximately 14 to 15 days. As you can see in the figure illustrated, I added stars to demonstrate where E. lateratus, what the shark was being fed, was first detected on the swabs. The results illustrated that the amount of sequence reads per participant in each sample varied, but for all 3 individuals, it grew near the conclusion of the experiment. Figure 2 shows the prey variety based on DNA analysis from cloacal swabs taken from each individual bull shark. This figure's x-axis indicates each individual juvenile bull shark collected, while the y-axis reflects relative sequencing abundance. The results indicate that prey DNA was found in 18 of the 21 cloacal swabs, excluding sample 2, 10, and 17, while host DNA was found in all cloacal swabs from each juvenile bull shark. Figure 3 shows the DNA gathered from all 21 juvenile bull shark samples. This diagram is also known as a cladogram, and it depicts the prey species diversity throughout all the cloacal swabs obtained. According to the findings of DNA analysis, the 21 cloacal swabs provided a total of 1,850,298 sequence reads. According to the sequence read analysis, the Aridae family was allocated to the majority of prey sequencing reads. These are also the catfishes. Mugalidae, mullets, was the second most common prey family, and were followed by the Serenidae, also known as sea basses family. These three families accounted for the majority of the recovered diet, whereas the other unique ESVs accounted for only 3% of total sequence reads. Figure 4 depicts a species accumulation curve demonstrating prey richness as a function of the number of juvenile sharks collected, with an emphasis on the ESVs, also known as exact sequence variations. Here, the x-axis reflects sample size, whereas the y-axis denotes ESV richness. Because the curve has yet to reach a plateau, and the slope of the regression line through the last 4 data points is greater than 0, the diet species accumulation curve indicates that 21 samples are insufficient to fully capture the species richness of juvenile bull sharks' diets in the system.
Discussion. The discussion of this article focused on 4 main points. First, they validated the use of swab fDNA. Next, they talked about these methods used under the field environment. And then methodological considerations were discussed prior to the limitations of using fDNA metabarcoding. Validating swab fDNA. Environmental DNA contamination is a big concern when considering diet analysis methods of marine species like sharks. Fortunately, the fDNA method developed for this research makes contamination of eDNA unlikely. This is supported by getting only about 33% of eDNA from digested species. In the case of contamination, we would expect most, if not all, to be from eDNA. In addition, the DNA of 3 Teleas species found in the eDNA should have also been found in swab samples if contamination did actually occur. The onset of prey DNA detection in swabs was previously described as 12 hours. In this study, DNA detection ranged from 3.5 hours to 2 days following sharks' first fillet acceptance. This may be because this study used a swabbing method compared to theirs, which collected the fecal matter directly. Or it could simply be because individuals have different retention and digestion times. Methods under field conditions. A major goal of this study was to assess the execution of sampling in the marine environment. Overall, it was successful because there was recovery of sequence reads from most of the tested sharks. The metabarcoding results were also consistent with previous diet data for species in size range and other systems that are quite similar. Doing this study under field conditions showed that fecal matter gives high taxonomic resolution. It is higher resolution than a method like morphology analysis of the shark's stomach content. Having higher resolution means it reveals a higher prey diversity, and evidence previously showed that metabarcoding found almost twice as much species richness. Methodological considerations. Overall, this method was easy to use, which is important when providing a new approach. It is not very invasive as it simply involves swabbing the sharks. And compared to collecting stomach contents and tissues for stable isotope analysis, it is a lot less time-consuming. The positive prey DNA recovered indicates that it is comparable, if not better, than other diet sampling approaches. Finally, this method has the potential of resolving trophic relationships that is so important and difficult to do in marine environments. Limitations of fDNA metabarcoding. Like every approach in the science world, there are some limitations. This method doesn't allow prey consumed by prey or incidental ingestions to be distinguished. It also can't determine prey size or life stage. However, there is some progress towards quantitative analysis of consumed items that looks possible in the near future. Specialized primer sets are required to target prey groups, and fDNA metabarcoding is sensitive to the time of defecation, so there's only a short window. Furthermore, there needs to be understanding on digestion rates in order to get accurate representation of taxa. So we need to know the time frame of when consumption occurred and for how long it was actually detectable. However, corrective factors can be used to estimate this. Future applications. This study is important in terms of future use. It provides evidence that fDNA metabarcoding can be a viable option for developing diet information in marine environments. So how can we improve this study? Well, there are two main ways we should improve this study for the future. The first being experimental design. The experiment can be manipulated to be used on larger marine vertebrates to see if the results can be generalized by recovering their prey DNA sequences. In addition, experiments that investigate the effects of mixed diets on fDNA recovery and composition would help to improve the study. Other things that could be done is verifying correlation between short-term feeding and biochemical tracer values of slow turnover tissues, which can be done by applying both of these methods to the same individual. Also, improving understanding of relationship between food retention time and fDNA signal decay would help improve the interpretation of these results. Now, the second way of improving the study is improving our sequence libraries. These experiments require lots of help from databases, so having good sequence libraries is vital. Region-specific fish barcoding databases will need improvement of resolution to help these molecular diet structures. Future research. fDNA metabarcoding is a method that should be used again in future studies. Using it can give us insight into marine environments that are typically hard to observe. In particular, we can better study marine trophic relationships that are not currently well studied. A future study should be conducted looking at non-sharks to see how well the results can be transferred to other marine species. Overall, it is a useful method in the field of molecular ecology that likely has many uses going forward.